Hello, and welcome back to part 4 of the lecture on chapter 10, Controlling Microbial Growth in the Body. Where we left off with the part 3 of this chapter was the different mechanisms that we were interested in of how to inhibit microbial growth. And so that was the end of part 3. And now we're going to transition to thinking in general. What are we looking for in an ideal antimicrobial agent? So we went over like the different mechanisms that they can use, but in addition to how they kill the microbe, what other features are beneficial or helpful to evaluate in an antibiotic or an antimicrobial? First thing is that it's readily available. It's easy to get. That it doesn't cost very much to produce, so that the medication cost is reasonable. Having something that's chemically stable is key because we do try to keep these medications on the shelf in the pharmacy. And if they need to be refrigerated or they need some other kind of special handling, that just increases the complexity of getting it to the market properly, but also increases the cost of the medication. So something that is shelf stable. Easily administered. So something that you could prescribe or that the patient can take in a way that maybe doesn't require another person. So oral medications, topical medications, those are easily administered. They can be administered by the patient themselves. Some medications must be administered intramuscularly, that is with an injection into the muscle, or can be administered intravenously, that would be into an IV line, which would need to be done in a hospital setting or some kind of clinical setting. Those are less easily administered, but sometimes necessary. Since we're looking for everything, non-toxic, not having a lot of side effects for the patient. That is this idea of selective toxicity. It's something that's going to target the microbe and not affect the patient. And then if on top of that, we could have something that doesn't trigger allergic reactions in people, that would be great. The two kinds of drugs that trigger allergic reactions that are in this category of antimicrobials, penicillins, the beta lactams, so people can have allergies to those, and the sulfa drugs. Also, there are other kind of niche allergies that develop, but those are the two big ones. And then selectively toxic, remember that means that it targets the microbes and not the human. But in some sense, we want it to be toxic against a wide range of pathogens, but there's a limit to that, and I'll explain on the next line. So what we refer to as spectrum of action is the number of different pathogens that a drug can be taken let me say that again. The range of microbes that are going to be affected by this particular drug. Okay. If we say the drug is narrow spectrum, that means it's effective against a few organisms. And in some ways, that's desirable if you can identify what the microbe is and identify it quickly. If you cannot identify it quickly and the patient is very ill and we know we just need to start killing microbes, we don't know exactly what this is, let's just let them have it, then that patient gets prescribed a broad spectrum antibiotic or a broad spectrum antimicrobial that's going to affect a lot of different things. There are some problems with that, but at least we can start getting the patient treatment, and then we'll deal with the problems later. And here are the problems. If you use a broad spectrum antimicrobial, it's effective against many organisms, including the desirable microorganisms that we have living on our bodies and in our bodies, in the digestive system especially, but also, for example, in the vagina. There's this delicate balance between bacteria and fungi. The Candida albicans is the fungus that's most commonly present in the vagina. This is a yeast, and it's supposed to be there. Between the bacteria and the yeast, they keep each other in check. They have what's known as microbial antagonism. That means those microbes are competing with each other for resources, and they keep the population of each one they keep the other one down, that is beneficial. So when a patient is prescribed an antibiotic that probably wipes out the whatever the strep infection or whatever it is, that other infection they have going on, but it is also wiping out some other desirable bacteria. Sometimes the fungi at that moment go, woohoo, party in the vagina, because they can get an increase in their numbers. That's what we refer to as a super infection. A secondary infection is when another microbe comes in from outside and creates an infection. Super infection is when you were already colonized, and that's okay. But then when we like killed off the local microbes inadvertently, that allows for the super infection. And so again, killing off the normal flora reduces microbial antagonism. This is why sometimes we see in hospitalized patients who have been on antibiotics for a long time, the development of something called Clostridium difficile, or C. diff. What happens is we kill off the normal flora, kill off at the same time that we're killing off the infection. And then if the patient gets colonized by C. diff, which we can usually tolerate as long as we have other bacteria present, there's no microbial antagonism. There's nobody home to protect the house. So think of it like guard dogs, that are protecting the property. If they're already there, they're already stationed there, and they're not doing damage to the property. They're just like helping keep other dogs out of the property. That's beneficial to the patient. And so if we kill off the normal flora, we lose this microbial antagonism and sometimes get colonized by less desirable microbes that take over. All right, so let's see a visual of spectrum of action on this next slide. This is selected spectrum of activity of selected antimicrobial drugs. And what we have on top are the different categories of microbes. We have the prokaryotes over here on the left, and we've separated them into these categories. So gram-positive are the ones we're most familiar with, gram-negative bacteria. But there's also mycobacteria. These are the Mycobacterium tuberculosis. They cause the lung infections, and they can go all over the body, but where we usually see tuberculosis first is in the lungs. And chlamydia, this is a sexually transmitted microbe. Let's see, the eukaryotes, the fungi are the ones that we're usually concerned with. There's some protozoan parasites and some worms. The helminths, or the worms, are sometimes referred to as the parasites. And then finally, the viruses. These are acellular. And no huge surprise, things that are going to treat viral infections are going to have zero impact on fungal infections and zero impact on bacterial infections. And likewise, the medications that are going to treat bacteria are going to have no impact on viruses. And this is one of the things that is most challenging for healthcare providers and for nurses to try to explain to patients. They come in with a cold and they want antibiotics, and the antibiotics are not going to help them at all if what they have is a viral infection, and in fact can lead to the development of antibiotic resistance, which we'll talk about shortly. But let's focus in for a moment on the prokaryote side of things. A few medications that we've already mentioned, for example, the penicillins. These are the beta-lactams. These are going to be, remember, they target the formation of that cell wall, and it's easiest for those beta-lactams to impact the gram-positive bacteria. Because the beta-lactams are right on the outside, and they're impacting the putting together of those NAM units. Notice that some of the beta-lactams, some of the penicillins, can also impact the gram-negative bacteria, but they have to get through that outer membrane. Remember, the gram-negative has the outer membrane, then a little cell wall, peptidoglycan, and then their regular plasma membrane. So if this molecule is small enough to slip through the pores of that outer membrane, then it might have an impact on some of the gram-negative bacteria. Okay, polymyxins, we said those impact the cell membrane of these gram-negative bacteria. And then you've got some broader spectrum antibiotics like the erythromycins. Erythromycin, let me think, we said that impacts protein synthesis. The tetracycline, same deal, different mechanism, but also impacts protein synthesis, and it's going to have an impact on all these different kinds of bacteria. The sulfonamides, those impact the metabolic pathways. It's a broad spectrum in the prokaryotes, but also some of the protozoa can be affected by the sulfonamides. And let's see, streptomycin, we said this one also impacts the protein synthesis. This one, that a different mechanism, it changes the shape of the ribosomal subunits. So this one affects, it has an impact on the gram-negative bacteria and the mycobacteria. And then we mentioned isoniazid. This one is one of the medications. It's one of the famous medications that's used for treating tuberculosis. And tuberculosis is, as far as bacteria go, very, very slow growing. And these medications, they work on the developing bacterium. In other words, they are only effective during that time when the bacteria is actively reproducing. And so these medications have to be given for months, if not years, to clear up an infection like tuberculosis. But it's going to have an impact on the tuberculosis and not on the gram-positive bacteria that, you know, are in the patient's body. Okay, I think that's everything I needed to go over with this slide. Now, how to assess how effective an antimicrobial is. There are 4 different tests we're going to talk about, and the third test is a combination of the first 2. So the first one is generically known as the diffusion susceptibility test. It's the one you've probably seen. It's on an agar plate, and little circles of paper that have been soaked in different antibiotics of different concentrations are put on that agar plate, and we can see the growth. I'll show you a picture. The older name for it is the Kirby-Bauer test, and you'll still hear people call it the Kirby-Bauer test. The second test is known as the minimum inhibitory concentration test. And as the name implies, what we're looking for is what's the least amount of medication that can be given that's actually going to impact the growth of this bacterium. Sometimes you'll see an MIC test, minimum inhibitory concentration. That one's done in test tubes. Then clever folk kind of put those 2 tests together into one test known as the E-test. So it's done on an agar plate, but it has the different concentrations of the same antibiotic. I'll show you a picture of that one. And then finally, kind of following up on this minimum inhibitory concentration test or MIC test, we have the 4th one is a minimum bactericidal concentration. And I'll show you how you use the tubes from the minimum inhibitory concentration test to figure out, okay, so that's the minimum that's going to inhibit the bacterium. What's the least I can use to really just kill them, completely kill them, gone? All right, so let's do them one at a time. Here's a susceptibility test. There is an agar plate. It has a bacterial lawn, so it's been streaked very generously with the sample of this bacterium grown up in the lab, and then these are the little circles that are soaked with whatever antibiotic are in there. And as you can see, there's visually an indication of this area where this solution that the paper disc was soaked in, that has diffused out into the agar, and it inhibits the growth of the bacterium all the way around. So the zone of inhibition is where that open area is. And so the larger the open area, the more effective that particular antibiotic. As you can see, if you this one and this one, these 2 medications would be worthless for fighting off this particular bacterium. And so this is how healthcare provider would send a specimen to the lab. We as nurses would be the ones collecting the specimen. Then it would be a lab tech who would spread it out on an agar plate, and then see which of these antibiotics would be recommended. This is going to take a couple of days to run, though, and so depending on how sick the patient is, an antibiotic of some kind might be prescribed first, and then a few days later, the orders for the antibiotic might change to something more specific based on the results of this test. Okay, then the second test, the minimum inhibitory concentration test, MIC. As I mentioned, there's a bunch of test tubes, and this is an eyeball test. You can just see, these tubes are still turbid. That means that there's a bunch of cells floating around in there. These are bacterial cells that are reproducing, the turbid tubes. And then you've got some clear tubes. If light shines through it, you have a decreased concentration or decreased population of microbes in here. And across the two, so the lower the concentration of the medications over here on the left side, and then as we move across through these different tubes, we have higher and higher concentrations of the medication. And what we're looking for is what's the least amount of medication that is going to keep this from growing very much. Okay, that's minimum inhibitory concentration. Eyeball, you can say there's clear, but we cannot guarantee that there aren't bacteria in here. So we'll get back to that point. Now let's see how you put together the Kirby-Bauer test and the minimum inhibitory concentration test into one test. And that is the famous E-test. It combines the aspects of those two other tests. So you're working on an agar plate again, but this time you're only looking at one antibiotic. But on this strip of paper, we have differing concentrations of the antibiotic. And so you can see there's a zone of inhibition, and then clearly at this very high concentration, huge decrease of bacterial growth or inhibition of bacterial growth. And then as the concentration gets lower and lower and lower, we can see there's, okay, if you go too low, you're not going to get inhibition of growth. But you get the idea. Okay. And then for the fourth test, we're going to go back and have a look at those tubes from the minimum inhibitory concentration test. These tubes again, remember we said, okay, this tube is for sure inhibiting the growth. So we're only going to look at the clear tubes. And we'll take the first clear tube, and I know the numbers don't match up with this image, but you get the idea. You take the first clear tube and pour some out onto an agar plate. The second clear tube, pour that one out. Third clear tube, pour that one out. And what you're looking for is any kind of bacterial growth down here. And in that first clear tube, yeah, it looked clear to the eye, but when we poured it out on the plate, we still got bacterial colonies. That means this is not completely bactericidal. It's inhibitory. It decreased the number of colonies, but there's still colonies here. And so we want to bump up one more step in concentration because that concentration is where we're not going to get any bacterial growth. And you might say, why would you bother to take the time to do this? Why don't you just go all the way to the end tube and know for sure we're not going to get any growth there? Well, for two reasons. Medications are not without cost and they are not without side effects. And so the less medication that is needed to treat an infection, the better off we are. And so that's why we look for the minimum inhibitory concentration. We say, okay, I know it's going to be close to this, but we're not there yet. Let's go up a step. And then we know that this one is bactericidal. This concentration is going to kill off the bacteria to the point that we're not going to have that bacterium growing. Okay, so that's the MBC, or minimum bactericidal concentration test. And that is the end of part 4. I will see you over in part 5.